going to be our last lecture for the school year. Woohoo! The crowd goes wild. And we're going to finish off with photosynthesis. Now, I know we've talked about photosynthesis throughout this unit, and it's something you've learned about in middle school and probably elementary school to some degree. So we're going to get probably the most in-depth with it you ever have before. But it's still not too bad, trust me. So I think this will be good to help clear up any misunderstandings that I was noticing in your mesocosm lab reports, and also just in understanding the carbon cycle relationship a little bit more. So here we go. 2.9, photosynthesis. So understanding 1, what is photosynthesis? So the definition, or what we would say, is it's the conversion of light energy into chemical energy. Chlorophyll is the main photosynthetic pigment that's going to be doing this. Now, if you remember back to when we talked about organelles, chlorophyll is a filling, a green filling or a green pigment inside these little pancake stacks, inside the chloroplast. So here's the word right here. Chloroplast is the organelle that are inside only plant cells, so not in animal cells, only plant cells have these, and they are what produce energy for plant cells. So remember, plants can make their own energy. We can't, okay? We have to eat something to get our energy. So what plants do is they take in carbon dioxide from the atmosphere, so you can see the equation right here, I'm touching it. They also take in water, usually through the roots from the soil, and then they need light from the sun, and that is going to create sugar or glucose for themselves, and they're going to use that sugar for survival. And they also produce oxygen, not for themselves, but as a waste product, so they release that oxygen into the atmosphere. So there's an inverse relationship usually between carbon dioxide in our atmosphere and oxygen. So when trees are doing a lot of photosynthesis, or in an environment where there are lots of trees, you might see a reduction in the amount of carbon dioxide in the atmosphere, and that's a good thing because that's good for the global warming effect or the greenhouse effect that could be getting too intense. So it's good to have a reduction of carbon dioxide, and also it's producing oxygen for animals as a waste product. So you might see O2 levels being higher when CO2 levels are lower, or you might see the opposite where CO2 levels are high, meaning there's not a lot of plants doing photosynthesis, and then you're going to see oxygen levels be a little bit lower in those areas. Understanding 2 and 3 have to do with the spectrum of light. So I'm going to break this whole image down. So we're going to start at the top with these wavelengths here. So these are called a wavelength. These are longer wavelengths because there's a bigger distance between each oscillation. We would call these an oscillation when they go up and down. So there's a bigger distance here between these two peaks, where here, if you go, these would be short wavelengths. They go up and down really fast. There's a shorter distance between each oscillation. So why I'm bringing this up is because if you notice down here, there's a scale that says 700 nm. These are nanometers. So this is a distance between these wavelengths. So longer wavelengths are on this side. That's hence the larger number. And these would be shorter wavelengths over here, 400 nanometers. So as I'm talking about this, and I talk about long wavelengths and short wavelengths, it has to do with the distance between their peaks, okay? So here we go. So when we talk about plants doing photosynthesis, they interact with the visible spectrum of light that we can see. So the sun there's a lot of different types of wavelengths, radio wavelengths, X-rays, gamma rays. The ones we talk about the most are usually infrared wavelengths and ultraviolet wavelengths coming from the sun. So you can see here there's a measurement for the range that they fall in. And we're just going to look at this tiny little rainbow strip here, and it's blowing up to be bigger for you. This is our visible spectrum of light. These are the colors that we can see with our eyes. So light from the sun is composed in a range of wavelengths. We can see all these colors. what we see are the colors that are not being absorbed by something. So with plants, for example, we see green. We see this color here on most plants because they are not absorbing that color. They're absorbing all these other colors, red, orange, yellow, blue, violet, but not this area of green, and so that's why we see that color. Same with a lot of other objects around us. When we see a white surface, it's absorbing all the other colors but white, and that's why we're seeing white. Okay? So when we talk about plants doing photosynthesis, we talk about chlorophyll. So remember, that's the pigment inside a chloroplast that is going to be doing photosynthesis. And it's a green pigment, usually, is what we would say. So chlorophyll A and B are the most common ones. Chlorophyll A and B like to absorb red, so these would be longer wavelengths, and blue-violet colors, which would be shorter wavelengths, the most. Okay, so that's why we see green, because they're absorbing those colors the most. Green, which falls into the 500 to 550 nanometer length, so it's kind of a middle range wavelength, is transmitted or reflected by chlorophyll. So that's why we know if chlorophyll is being green, because that's what we see, but now we understand that it's not because it is green, it's more that it's absorbing all the other colors of the visible spectrum of light. All right, so this is one of the skills. You could be asked to draw an absorption spectrum or an action spectrum, and this goes with chlorophyll A and B and the visible spectrum of light. So I'm bringing it up here. So it's skill 1, absorption versus action spectrum. So an absorption spectrum shows the quantity of each wavelength of light absorbed by a specific pigment. So if you look here, this first graph is an absorption spectrum. So you can see the y-axis is measuring the quantity of absorption. Okay? And here is the different colors or wavelengths that it's absorbing. So chlorophyll A is this light green line. Okay? Chlorophyll B is the darker green line. And then there's also other pigments out there that absorb. So a carotenoid, which is this orange line, is a different type of pigment that plants might have. It often is what makes plants look an orangish color or a orangish pinkish color. So when you look here, you can see that chlorophyll A, its absorption level is higher in these colors, so violet blue, and over here in this orange red color. Chlorophyll B is slightly different. You can see it likes blue a little bit more than violet, and in the shorter wavelength category, and in the longer wavelength category, it likes the orangish more than this reddish color. And then for carotenoids, you can see it likes the blue spectrum the most for absorption, for pigment. And so you can see that the areas that all of these are not absorbing are falling into this green-yellow stage here. Carotenoids a little bit more over here it's not absorbing green, yellow, oranges, and reds. Right? So that's why you're going to see a lot of times yellow, orange, and red things coming from something that has carotenoid in it as a pigment. Where with chlorophyll A and B, notice that it's very flat in this green-yellow section. So you might see plants looking more green and yellow that have those pigments. So remember, what we see is what's not being absorbed. So an absorption spectrum showing you what's being absorbed versus not. An action spectrum is the summation of the individual absorption spectra of the various pigments. So it's showing you the relative effectiveness in photosynthesis. Its action is how well is it doing photosynthesis with these different pigments inside of it. So this is an example of an action spectrum. So the action is how fast is it doing photosynthesis, the rate. And you can see here that this plant is doing better because it's absorbing these colors more. Violet, blue, and green is when it does more photosynthesis. It is not doing photosynthesis at this point when it's looking at green and yellow colors. And when you go back over into the orange and red, it has a higher rate of photosynthesis. So this is an action spectrum. So y-axis is the rate of photosynthesis, and the maximum rate, which would be these peaks here, are at the blue end and red end of the visible spectrum. So it tells us what plants will do photosynthesis better if they have pigments that absorb these lights, these colors. Okay, so now we're going to get into photosynthesis. So this picture has a little bit more detail than you need, but what I like about it is that it's all in one place. So the main thing you need to know is this equation up here. So carbon dioxide plus water plus light is going to create sugar and oxygen. This happens through two steps. So this is a chloroplast right here. There's two reactions that occur in photosynthesis. There's the light-dependent reaction and the light-independent reaction. So think about it as light-dependent means it needs light in order to happen. So you should be able to assume that this is where the light part of the first equation comes in. And then the light-independent reaction can happen without the light. So but one has to happen before the other. So the light-dependent reaction has to happen first. It also shows you where the other characters come into play. So notice water, which is part of the equation up here, is going to be used with the light in the light-dependent reaction, and it's where it's going to produce that waste product of oxygen. So a lot happens in this light-dependent reaction. The light-independent reaction is where the carbon dioxide comes into play, and you're going to be producing the sugars. So let's get into it. So understanding 4 is, let's get started on how photosynthesis works. So we're going to first focus on the light-dependent reaction. So that's going to be happening here in the chloroplast. So this is supposed to be a bunch of pancake stacks here that looks like a chloroplast. So energy is absorbed by chlorophyll, and the protein complex is used to produce ATP. So here you can see the light is going in, and it's going to need to make some ATP, some energy, in order to get the light-independent reaction to happen. So it's important that the light reaction or the light-dependent reaction makes that ATP. It happens through a process called photolysis. Photolysis is what it is basically the splitting of water with light. Anytime you see the word photo in front of it, it means light. So light and then lyse means to split. So light splitting. So photolysis is going to split the water molecule, and what you're going to get in the end is a bunch of hydrogens by themselves and some electrons that are going to be used to make energy, the ATP, and then some oxygens. Those oxygens are going to combine with each other and create O2 and be released out of the chloroplast into the atmosphere. And then those electrons are going to be used with the hydrogen to make ATP, which is going to start the next cycle, the light-independent part of the reaction here. So photolysis is that first you need the light and water. You split it, you're going to come out with, here's the equation. The water is going to be split into 2 hydrogens, 2 electrons, that's what that E stands for, plus 1/2 of O2, so 1 oxygen molecule. In this picture, you can see it happening as well. The light's coming inside the chloroplast. This is 1 chlorophyll stack right here. And the water is going to be split. You're going to get a bunch of hydrogens, some electrons that'll be used for the light-independent stage, and then oxygen molecules released out into the atmosphere. So that is the first stage of photosynthesis. It's the light-dependent reaction. Now let's talk about the second part of photosynthesis, and how it works is the light-independent reaction. So what's going to happen now is the carbon dioxide is going to be used or absorbed from the atmosphere, and it's going to be changing from inorganic carbon dioxide, and it's going to become an organic form via fixation. So what fixation basically means is carbon molecules combining together to make sugar. So, or anytime you see fixed, like when it's carbon fixation, it means multiple carbons being added together. So in this case, it's going to be making sugars, because sugars, if you remember, if we just look here really fast, is C6. There's 6 carbons involved in this. to make the sugar. So inorganic carbon dioxide molecules will become organic form via fixation. It's going to make sugar. And it's going to use hydrogen from the photolysis and ATP in order to make this process start. And in the end, you're going to come out with sugar. You do not need to memorize this Calvin cycle here. Calvin cycle is another name for the light-independent reaction. They're the same thing, but basically it's showing you in more detail how it ends up being the six carbon molecule, this glucose. Okay, understanding 6. What factors affect the rate of photosynthesis? So there's a couple things that could affect how fast photosynthesis happens and how efficiently it happens. So we have 3 limiting factors here: temperature, light intensity, and carbon dioxide concentrations. So temperature, here's a graph representing this and how fast the rate of photosynthesis is. There is a perfect temperature and there are enzymes involved in this photosynthesis process, and they can become denatured if it gets too hot. So notice that there is a perfect temperature for a very efficient rate of photosynthesis, and anything on either side of it is going to decrease the rate of photosynthesis. A warmer temperature increases molecular collisions, so this is why it becomes more, it occurs more quickly. Light intensity. Visible light, especially the red and blue light, is essential for activity of chlorophyll during the light-dependent reactions. So in this graph, you can see there is a positive correlation between light intensity. So the increase in light intensity increases the rate of photosynthesis. And then enzymes are already working at their maximum rate at this point if it has a good light intensity. And eventually it plateaus out. This is showing carbon dioxide concentrations. Carbon dioxide is essential in order for the light-independent reaction or the Calvin cycle to happen and produce those sugars. So you can see a positive correlation as well here. As there is an increase in carbon dioxide concentration, the rate of photosynthesis increases until it eventually plateaus. So the question here is, how would you, if you were to do an experiment, how would you test these factors in an experiment? How would you prove that these are things that could be limiting photosynthesis? So if you wanted to do something like this for your IA, these are some examples of things you could do to test. You could test O2 production levels, and you could do this by placing a plant in an enclosed tube, and we can use a veneer probe, and we could measure the amount of oxygen over time, because oxygen is going to be produced by the photolysis of water during photosynthesis, remember, from the light-dependent reaction, and we can see how much oxygen is produced, and we could figure out the rate of photosynthesis. Or you could do kind of something similar with carbon dioxide uptake, so how much carbon dioxide is being subtracted from the air around it. So you could place a plant in an enclosed volume and monitor the changes of CO2 with a veneer probe that's being stuck in the tube. Carbon dioxide is consumed during the Calvin cycle, so you should actually see a decrease if photosynthesis is happening. Also, you could do it with plants. You could have plants inside some water, and you can check the pH of the water, because the more CO2 dissociating into water, it becomes bicarbonate, and this makes the pH increase. So you could be checking for a high pH level, and then you would know carbon dioxide or photosynthesis is occurring in those plants. You could also look at biomass and how much of the biomass there is present. So you could measure the amount, the organic content of plants before and after an experiment. So organic content of plants is usually measured by removing water through dehydration in an oven and then weighing them afterwards. So you know how much it's actually not water, but is actually carbon. So these are some things you could do if you were interested for an IA when you get to that point, and how you could do it. So one of the applications you need to understand about why photosynthesis is so important is because we would not be able to, and many animals would not be able to be here on Earth without photosynthesis happening because we needed oxygen in our atmosphere. So what you need to understand is how Earth's atmosphere and oceans and rocks have changed because of photosynthesis. So just to give you a little history, the primordial Earth had a reducing atmosphere that contained very low levels of oxygen, so only about 2%. Then there were these little, what we believe happened through evolution, is cyanobacteria, which are little prokaryotes that do contain chlorophyll, were the first ones to be able to perform photosynthesis about 2.5 million years ago. As they were able to do photosynthesis, this created oxygen gas as a byproduct, and it was starting to be released in the atmosphere. Oxygen levels remained at about 2% until about 750 million years ago. And then from that point on, we saw a significant rise to what we know today, which is 20%. So we believe these little guys, cyanobacteria, were the first ones to start putting oxygen in the atmosphere. Oxygen generation also allowed the formation of our ozone layer. So the O2s they dissociate because of heat, and then an extra oxygen, after an O2 is broken apart, you now have singular oxygens. They bond with another O2, and that creates O3. What ozone does at very high levels in our atmosphere, meaning like higher in the atmosphere, like higher altitude levels, the ozone shields the Earth from damaging levels of UV radiation, and this in turn can lead to evolution of a wider range of organisms because many organisms couldn't handle the intense amount of UV radiation. So with oxygen now in the air and an ozone protective layer, many organisms were able to flourish and evolve and come to life. Also within our rock layers, iron compounds in the ocean were oxidized, meaning anytime you see the word oxidized, it means bonding with oxygen. So the insoluble iron oxides, now because they are bonded with oxygen, became precipitated on the seabeds, and over time and further sedimentation produced rocks with layers of rich iron ore inside of them. So we were able to get iron. And then this last part here, oxygen in the atmosphere has led to the production of other oxide compounds, for example, carbon dioxide, and how it oxidizes in oceans. So that's a little history of how important photosynthesis is to the atmosphere, oceans, and rock depositions. Okay, so one of the skills I told you would have to be able to do is drawing an absorption spectrum, and an action spectrum. So remember, absorption spectrum has to do with for chlorophyll. So it's going to show you each pigment individually and how much is absorbed at each wavelength, where in this case, this picture is showing you an action spectrum for photosynthesis. So here you can see on the y-axis, it's the percent up to 100 of the maximum rate of photosynthesis. And here you're seeing the wavelengths from short to long. So remember, violet and blue is usually in the shorter category, where orange and red are in the longer categories, and it goes in order like a rainbow. So violet, blue, green, yellow, orange, and red. Notice that it's less photosynthesis in the green, yellow, middle range category. Here is an absorption spectrum. It's on top of the action spectrum, so you can still see that action spectrum in the background. But here on this other y-axis, we're seeing the percent of absorption of light. So here you can see it's a little bit higher absorption of these colors. And over here, it's higher in these colors, in orange and red, and then flat along the middle wavelengths. So if you had to draw this, you would usually draw exactly what you're seeing right now. This is the pattern. So you can tell that having these pigments is going to allow for this rate of photosynthesis to occur. So higher rate of photosynthesis on either end when you have these colors being absorbed by pigments. So this might bring up the idea of why do leaves change color in the fall, or what's happening there? Why are we no longer seeing green and we're seeing orange? So here it's saying, do all plants have the same action absorption spectrum? This is definitely not true. For example, this is a different species it's a water type plant. So, and notice that it absorbs, so here it says percent of absorbed light, and this is on the same on either side. Notice that it actually is absorbing those middle green wavelengths. So it's absorbing all of them. and you're ending up with this brown type color that we're seeing. Here it's saying what we blame this on is accessory pigments. This is so not all species just have chlorophyll A and B and carotenoids. So accessory pigments allow some plants to take advantage of the green wavelengths of light. And it's a competitive advantage in low light conditions such as underwater. So this is a water species. And so it actually can absorb some of those green wavelengths so that it can try to have more of a chance to do photosynthesis more efficiently. Why do leaves turn brown in the autumn? Some leaves have accessory pigments which cannot be seen when chlorophyll is active. So when the seasons start to change, chlorophyll usually breaks down, and it'll come back in the spring and summer seasons. But in the fall and winter, it starts to break down, and the accessory pigments don't break down as fast, so they start to show through. So we start to see some of those other colors, those other pigments in the plant. So we start to see those reds and oranges that usually are hidden because the chlorophyll is so strong and it's absorbing all those colors except green. So we start to see the other pigments, and so this orange leaf here, what you would assume is that it does not absorb that orange pigment, and that's why we see it. It's absorbing green pigment wavelengths. So that is why we see different colors in plants, is because of these accessory pigments. Okay, so normally, and we are going to do a lab, but normally we would do a lab called chromatography. So here, this is chromatography paper right here. And we would look at different plants and we would see what pigments are inside of them. So we're going to do an online version of the lab, but I promise you when we get together again in the future, we will do this lab, because I want you to have the physical hands-on experience with it. But this online lab will at least help you understand the equipment needed. It's a super simple experiment, but it's really cool because you could do it with any plant around you at home if you have this chromatography paper. So basically what you're seeing here is we drew this pencil line here in this paper. And you're going to put down, for example, I put down lettuce, and I just take a coin and I put lettuce here, and then I roll the coin over this line back and forth, back and forth, back and forth. And then I put this paper in a test tube filled with alcohol, and the alcohol is going to trigger the pigments to start separating. So you can see there's pigments at different colors inside this plant. Okay, so you see the yellow ones, there's green, there's blue, and then there's yellow up here again. And we're able to figure out what different pigments are inside each plant, and it's pretty cool, based off of something called an RF factor, which is going to tell us what are the different types of pigments inside of it based off the distance they travel from this starting point. So we're going to do that on an online lab, but I promise you we'll do it with different plants when we get back. Okay, so we're not going to move on any further from there. And next class, we'll do the online lab. So this class, you just needed to take notes, and please make sure you put them into ManageBack when you are done. And I hope that was great and helpful. And if you have any questions for me, please put them in the chat while you're watching this Edpuzzle video. Last lecture of the year, you guys. You did it. I'll talk to you soon.